Hi, hello everyone. It's my honor to be here and give this presentation in honor of Warner. So I arrived to the Warner's lab 4 years ago and as a postdoc, and I'm currently a research scientist here. So my title today is Mapping Genetic Interactions in the HIV latency toward an art-free remission. So what is HIV latent reservoir? I just Googled it earlier this week, and the top hit is from an NIH website. It says a latent HIV reservoir is a group of immune system cells in the body that are infected with HIV but are not actively producing new HIV. So as numerous works as here reviewed by Warner and Silicano revealed that the frequency of latently infected cells are actually very low. In its natural host, CD4 T cells, on average, it's only 1 in 1 million CD4 T cells. They have HIV latently infected in there. But because we have more than 1 trillion CD4 T cells in the body, so on average, for every person living with HIV, there's about 1 million latent reservoir in them. So 1 million cells, when you spin them down, is smaller than the tip of a pin, but when they spread in the body, in the bloodstream and the lymph nodes, they can cause big problems like bombs in a haystack. So we have marvelous drugs, mostly from Gilead, that can target HIV life cycle in different steps, but they all target different steps that is at lower sequences of the viral life cycle, including viral assembly and budding and infection of the new cells. However, currently there's painfully no drug to target the very upstream of HIV life cycle, the viral transcription. And because of this, the virus is always trying to get into the active transcription and get into the active life cycle. You have to keep the pressure of the drugs in there for all over the lives of the individuals. And Dr. Gallo mentioned earlier, there's a small population of people living with HIV. They are elite controllers. They never develop into AIDS, but they also never take the drugs. And for those elite controllers, no matter how they get there, and the recent breakthrough revealed that for HIV in those people, the HIV are mostly integrated in inaccessible heterochromatins, and they are far away from cellular transcription start sites, and they are covered by repressive chromatin marks. So this strong viral repression induced by those repressive histone modifications indicates that it is possible to epigenetically block HIV transcription once and for all, just to try to reproduce what is going on in the elite controllers. So, and in contrast to elite controllers, in regular people living with HIV, if we zoom into that promoter region, HIV, they prefer to integrate into the active chromatins in the human genome. So this makes their promoter region accessible to transcription factors such as predominantly NF-kappa B discovered by Warner, and also PTFB, which can promote the elongation state of RNA polymerase II, basically makes the POL2 travel faster and produce more HIV RNA. So, but the HIV latency is the result of a number of host repressors, many of them discovered here by scientists here and around the world, including those factors that inhibit NF-kappa B and the factors that inhibit PTFB by degrading it, and also factors that can directly bind POL2 and inhibit POL2 and induce POL2 premature termination. And also earlier, this decade, people found that the BAF BID4S complex can push the nucleosome onto the LTR to make the LTR harder to be transcribed. And so normally we also put a GFP reporter in the lab. We put a GFP reporter in the HIV genome and use GFP level to study the viral activity. So this gave us a chance to test the effect of individual host repressors on the overall HIV transcription. As we show here, when we knock down one of them, the GFP expression level may be higher because when we remove one of the players here, the HIV will have a higher chance to be reactivated. So the first question we asked is that, because if we want to learn from those host repressors, when we have a chance to increase their repressance and increase those epigenetic inhibitions, and we may have a chance to permanently block HIV transcription as in elite controllers. To achieve that, our first question we ask is, are there more unknown host repressors for latent HIV? How to get the whole picture of those potential factors controlling or repressing HIV expression during the latency. To achieve this, we constructed a platform called reiterative enrichment and authentication for CRISPR targets. Basically, it's a genetic screen using the CRISPR machinery. So we used CD4 T cells, and we sent D-Cas9 crab in those cells. And we also, in those cells, they have a latently infected HIV labeled by GFP. And then we sent in the CRISPR RNA library, and then we sort the GFP positive cells from a large population of these host cells. And then after sorting them, we extract RNAs from those cells. But instead of directly going to sequencing, we cloned them back into the original vector of those RNA, and we send them back into the original cells to repeat this process until we get significant GFP percentage increase induced by those RNA libraries enriched in the later rounds of screen. And we use this method because HIV had this problem of so-called stochastic HIV reactivation pioneered by Leo Weinberger here in Gladstone. And they found that even by chance, because of random things, HIV always has a chance to exit their latency and contribute to the active HIV reactivation. So this is the problem both clinically and in the lab, because in the lab, it can increase our chance of getting the noises as shown here, because even if you don't treat the cells with any RNA library, it already has a non-0% of the GFP label here. And if we sequence this, the library underlying this in those cells, we found very little hits. Those two hits are false positives. They regulate cell growth, but not HIV activity. So through this kind of iterative screening, we could get a library that progressively enriched of those RNAs that actually work to block, to active HIV transcription. As shown here, the later round of RNA library can react with HIV from about 16% of the cells. And the sequencing of those library, indeed, we very unambiguously identified a group of the factors that were later confirmed. Those are 7 top hits that can control HIV reactivation in the CD4 T cells. So those 7 top hits, as I listed here, 2 of them are known. Alpha-B alpha is involved in NF-kappa-B pathway, and CYLD is also involved in NF-kappa-B pathway. But the other 5 factors are relatively new. That was 4 years ago, so at that time it was very new. And as we can see here, that their functions are highly diverse. And I have some mechanistic data showing that they regulate either transcription initiation, elongation, or premature termination of the HIV. So this led us to ask, do these factors synergize with each other? Because if they target different pathways and you combine them, they will synergize. And we found indeed that, for example, here, when we knock down PSMD8, which is a factor of the proteasome that regulates HIV transcription elongation, and when we knock down CYLD together, CYLD, remember, is involved in NF-kappa-B pathway. The combined knockdown can much more strongly increase HIV activation compared with knocking down them alone. So we see they are synergizing because the predicted additive effect is around here. And in contrast, knocking down alpha-B alpha and TMEM178A, which both regulate transcription initiation, there's no synergy between them, only additive effect. So this phenomenon led us to generate our second hypothesis, which is that there may be hidden host repressors that by themselves alone have no effect, but could synergize with known host repressors to inhibit HIV expression. So those hidden factors are important because those means those are underdogs by themselves, they are not effective. But knowing them will give us a chance to boost their functions, just to increase the natural host repression on HIV. To reveal those factors, we repeated genome-wide screening, and using this method we call REACT for synergy, or REACT-S. It's very similar as the first screening method, but in contrast, we first introduced in every cells, in every HIV-infected cells, we introduced a guide RNA called SD query. Those query guide RNAs targeting one of the 7 major factors that I showed earlier. And then on top of that, we introduced another guide RNA library called input. This input library, they have different selection markers on them, so I can guarantee that in each cell, it has one query and one input in there. And then we iterated this screen, but with this time we only focus on the input. I have PCR primers that can amplify the cassette of input guide RNA, but not query guide RNA. So through this kind of iteration, we conducted 7 screens using 7 guide RNA, query guide RNAs. And this is what the result looks like. And they give us a map of the synergies or so-called genetic interactions between host factors. As shown here, this map has 3 different functional groups based on their implied function from previous research. Some of them regulate transcription initiation, some of them regulate elongation or premature termination. And there are multiple ways to use this map, including synergistic drug combinations as shown here, but I will not go into details. What I will show you just a little bit today, due to the time limit, is that we found 3 new repressors for HIV transcription called BCL7C, Cancer2, and 32. And so one example is the primary data shown here is that these new factors, they can strongly synergize with known factors. For example, NF-κBA, which is an inhibitor for alpha-B alpha. When we knock down alpha-B alpha alone, it has a very little effect. It can increase HIV activity by about 4-fold. And when we knock down BCL7C alone, itself has no effect at all. But when the 2 guide RNAs are combined, the combined knockdown generates a much, much higher effect, 65% compared with knocking them down alone. So 65-fold. So, and my mechanistic study so far showed that these 2 factors, 2 of the new factors called BCL7C and Cancer2, they work somewhat similarly. They can, when I overexpress them in the cells, we found that their overexpression could increase a histone marker called histone acetylation. Histone acetylation on HIV LTR as shown here. And this histone acetylation in turn recruit a known repressor called BRD4S onto the HIV LTR. This BRD4S has a binding pocket on it that combines those acetyl groups. The excessive amount of BRD4S, as shown by CHIP here, by chromatin immunoprecipitation here, can lead to the stabilization of the nuke 1 on the HIV LTR and block viral transcription. And more interestingly, this BCL7C and Cancer2, they don't work alone. They themselves are known to form their own complexes called BAF complex and NSL complex. And our data also show that these 2 complexes actually they can interact with each other to form a supercomplex. We call it BAF and NSL supercomplex. Remember, this BAF complex is actually can actually increase the loading of nucleosome on HIV LTR. And this NSL complex, which has this Cancer2 subunit here, it can hyperacetylate the incoming nucleosome. So we believe that this supercomplex play a key role by both pushing the nuke 1 onto the HIV LTR and hyperacetylating it, which in turn recruits BRD4S to induce epigenetic changes that contribute to HIV silencing in those latently infected cells. And indeed, we found that those two factors are key in this complex, because when we overexpress these two factors in CD4 T cells from people living with HIV, we found that the level of viral activity decreased by about 50%. This is a log value, by about 50% in those cells when those cells were activated by PMA and ionomycin. So which indicates that those two factors are so-called bona fide repressors for HIV transcription and maybe targets for future drug discovery. So here I just give you one example of how to use genetic interactions to do HIV cure research. And the remaining questions include that along this line of research, we need to know the complete components of this complex and what is the histone modifications that attract BRD4S. We need to have a higher resolution of these kind of modifications. And how about the specificity? Our preliminary data indicates that there's about 46 human genes are also regulated by this complex. Some of them are involved in host immune response to viral infections. So before we target this protein for HIV, we need to know the immunological ramifications by targeting this protein by studying those 46 human genes. And last, we can look for drugs that target this super complex by designing some clever drugs screening platform, just looking at the drugs that can increase their interaction and their function. And overall, our long-term goal is to continue the genetic screens in the latently infected cells and possibly using more kinds of hosts, more kinds of subtypes of the T cells. And eventually, we are going to look for increased list of host factors, and most importantly, the pairs of host factors that synergistically regulate HIV, and use by studying them and targeting them, try to permanently silence HIV. Okay, so I will, to start, I will thank, I have a lot of people to thank here. To start, I will thank all the research participants from the HIV/AIDS community that made our research possible. I thank my mentors, colleagues, and friends in Gladstone and in this community that contributed tremendously to my research. And this research was first supported as a pilot research from CHRP, a grant to me, and right now it's fully supported by the HOPE grant and from NIH. And in the end, I want to thank Warner and for recruiting me and keeping me in Gladstone 4 years ago and over all those years. And I thank my former colleagues, and many of them are still my colleagues, especially senior lab members, Robin, Mauricio, and Nadia, and they imbued me with Warner's rigorous scientific DNA when I first came into the lab. And now, many of my colleagues, and including me, we are budding from Warner. We are separating, and we are just like apple leaving the tree and the seeds leaving the plant, and we are ready to spread Warner's excellent scientific DNA to other places and other research areas. Thank you so much for your attention. Thank you, Warner.